Thank you, Rachel, for housekeeping. And good morning, everybody. My name is Joji Muramoto. I'm a cooperative extension organic production specialist at UC Santa Cruz. And welcome to the second session of this organic nitrogen management workshop. Today, we're going to have three excellent speakers. And first, by Michael Cahn. He's going to talk about water management strategies on efficient use of nitrogen in organic systems. And then second speaker is Margaret Lloyd. She gave a talk last week, excellent talk on cover crops. And then today she's going to talk about estimating nitrogen availability in compost. And the final speaker today is Patricia Lazicki. And then she's going to talk about estimating nitrogen from organic fertilizers. Each talk is going to be 30 to 40 minutes. And then as Rachel said, we're going to have a Q&A session after each presentation. And I have a brief announcement for Spanish-speaking listeners. And from my assistant's gonna make announcement. Here we go.
Para hispanohablantes, sigue al fondo de la pantalla y allí encontrará el enlace para la traducción.
Thank you very much, my assistant. Okay. All right, so let me introduce the first speaker today. Michael Cahn is an irrigation and water resources farm advisor in Monterey, Santa Cruz, and San Benito counties with the UC Cooperative Extension. Michael has conducted extensive research on improving nitrogen use efficiency through precise irrigation management. He developed the online irrigation and nutrient management decision program, Crop Manage. With that, Michael, take it over.
Thank you, Joji. Okay, so I'll just be focusing sort of broadly on water management in the context of organic systems and nitrogen management. It's a broad topic, but hopefully I covered some of the main points here today. So the first place really to begin is, why is water management important for nitrogen management in organic system? Why is it critical? And I think last week we really got a sense of how mineralization is very important for the availability of the inorganic nitrate and ammonium forms of nitrogen for crop production. And nitrogen mineralization rates not only depend on, temperature, and we talk about carbon nitrogen ratios and things like that, but also soil moisture. It has to be optimal to optimize nitrogen mineralization, not too much water, not too little water. Also, when you think about it, water management really affects how that crop's going to develop. And a lot of the fertilizers, amendments we put in the soil are actually quite dispersed in the soil. It's not necessarily in bands. And so we need a strong, well-developed root system to utilize those nutrients. So water management also affects just having a robust crop and root system underground to use those nutrients. Then for those who rely on groundwater for irrigation or may have some nitrogen, use surface water that may have some nitrogen, we often have a significant contribution of nitrogen from our irrigation water. And then a very important factor is the form of nitrogen that crops often are taking up is nitrate. And so we're investing so much in having this pool of nitrate to keep up with the crop and uptake. But nitrate is very easily leached. So when we over irrigate, we lose that that pool of nitrogen. And we were talking, a lot about nitrogen budgeting, and we'll continue to talk about it, where we're looking at what's the crop demand for nitrogen as we go, through the crop cycle. Early on, for a short-term vegetable crop like lettuce, it's quite low, but then it rapidly increases to maybe 3 or 4 pounds of nitrogen per acre per day. And then we have to keep up with that demand with the supply, and that can come from mineralization from soil organic matter, crop residues, including previous crop and cover crop residues, organic fertilizers and amendments such as compost that we put in, and of course the irrigation water. So you can do a lot of nitrogen budgeting, but I would contend, if you're not paying attention to water management, that budgeting for the season is not going to be that accurate, because if you over irrigate, you're going to leach a lot of that nitrogen right out of the root zone. If you under irrigate also, you won't grow a crop that utilizes the nitrogen in that soil very well. So optimizing water management is really important for nutrient management. So some of the important strategies for optimizing end use efficiency in organic system are real basic. First, you really want to apply the water with your irrigation system as uniformly as possible, and then schedule irrigations really to match the crop water demand, because we have a lot of control over that. You don't have so much control, with nitrogen to make that available to match crop demand, but with water, you do have a lot of control. And then you want to monitor your soil nitrogen levels, nitrate levels in the soil to understand, when do you have high pools of nitrate in the soil, so you're very careful not to leach that nitrate. And then taking credit for the nitrate in your irrigation water. So these are the main strategies I wanted to cover in this half hour. In terms of monitoring soil nitrate levels of your field, you're probably quite familiar with the soil nitrate quick test. We've talked a lot about it in conventional production systems because you can always add some liquid fertilizers pretty cheaply. But we're trying to encourage, in that case, growers not to over fertilize. But in the context of organic systems, we want to know when we have sort of a larger pool of nitrate available, so that's a time of the crop production that you definitely want to be very careful with your water management. So an example would be when you're establishing your vegetable crop early on. Often there's been mineralization that's happened with amendments you've incorporated, and you could start to develop a pretty high pool of nitrogen in the soil. But that really is sort of a time when you might over irrigate in establishing the crop. So being aware as you go through the season of what that pool is of nitrate is quite important. You want to learn more about the specifics of using soil nitrate quick test. I encourage you to visit our blog article in the Salinas Valley Agriculture blog, and that has some links to different materials for using that test. So the distribution uniformity, or we say the application uniformity of your irrigation system, often affects how much water a grower or irrigator is going to apply over the season. And the way we measure distribution uniformity in a sprinkler system is using these low-tech buckets, and we just collect the amount of water that we get in these buckets, and we calculate the uniformity of the lowest quarter. So the way that works is we compare the average amount of all the buckets to the lowest amount in 25%, and the lowest 25% of the buckets, and we calculate a uniformity coefficient. So I just wanted to illustrate how that would affect the irrigation requirement. And so if your intention is to put on, say, a ½ inch of water, if you have a 90% uniformity, it means most of the area is getting the same amount of water, and you only have to apply a little bit more for the lowest quarter of your field to get that ½ inch. But if you have a lower uniformity, say a 50% uniformity, you'd have to apply an inch of water to the entire field to be sure that the driest quarter or lowest quarter gets at least a ½ inch of water. What that means is in a lot of the field, you're going to over apply water just to get that driest area to have sufficient moisture. And in that, those other areas, you will tend to leach the nitrate that's available in the soil. So just as an example, this is how mathematically we calculate an irrigation requirement. We look at the crop needs, the crop ET, and we divide it by our distribution uniformity. And so if over a cropping season, the crop needed, say, 8 inches of water, then with a 60% DU, distribution uniformity, we'd have to apply about 13.5 inches, versus with an 85% distribution uniformity, we only need 9.4 inches. So just a little bit more than 8 inches of water. So we're applying much less water, and we're providing the same amount of moisture to the crop without over irrigating. So potentially, with drip systems, we should be able to get a distribution uniformity of 85% to 90%, and with sprinklers, 75 to 80% uniformity. Over the years, we've evaluated irrigation system uniformity on the Central Coast. For drip systems, we just put these cups under the emitters. We isolate them with these little pieces of hose that we put on the drip tape. And we've done a lot of fields over time. And we noticed, for drip systems, sometimes they provide very uniform application. But on some cases, you can have very poor uniformity. So on average, it was a little less than 80%. These are in about 10-acre fields or 15-acre fields, with the highest at 96%. With sprinklers, we get about 71% on average. So both of these systems, we see room for improvement, especially in some fields where we find some really low values for uniformity. Now we have a lot of growers using drip irrigation, and one thing I want to emphasize is, really pressure management is the key to drip. Of course, maintenance and operating things correctly, but you really have to focus on getting the pressure right. Most drip systems are running at very low pressures, about 10 psi or 12 psi, and in some cases even lower. So having a little variation of just 2 or 3 psi, and when I'm saying psi, this is pounds per square inch of pressure, that will affect the application rate in those particular areas of the drip system. So getting the pressure right is very important, and then having it very consistent from irrigation to irrigation, because most people do their scheduling based on hours, and if on some irrigations you're only putting 1/10 of an inch on, and another one you're only putting maybe 1/1500ths of an inch on, each time there's big variation how much water you're putting on per unit time. So having very consistent pressure is also important. We noticed that some of the worst uniformities that we measure in drip systems have to do with operating those systems at too low of pressure. And just sort of a observation I've seen on the Central Coast is, for surface drip systems with very thin drip tape, Many irrigators want to operate at lower pressure because they have too many leaks. And you don't notice the leaks when you run it at lower pressure. So there is training you need with your irrigators to make sure they're operating things at the right pressure and keeping it consistent. One of the important tools to use in drip systems are these pressure reducing valves or regulators, because they can maintain a consistent pressure in the drip systems during the entire irrigation and between irrigations. So with each irrigation, you put on exactly the same amount of water. Now, a lot of growers have had bad experiences, I know, with pressure regulators. And one of the reasons is they may not be selecting the pressure regulator for the specifics of their drip system. So they need to be selected for the flow rate range and pressure range of that drip system. And they need maintenance. You see these tubes here, they sense the upstream, downstream pressure, and there's little filters in there. And if they clog up, they won't sense the upstream and downstream pressure. Also, I know that irrigators will sometimes not use the pressure regulators, even though the grower buys them and has them installed. They put them in the open position. And that's because they've had a bad experience, or they need training on how to make use of these pressure regulators. So definitely there's some training components that you have to add to integrating this into your irrigation. But I think these pressure regulators are really important for drip systems to get that consistent application rate from irrigation to irrigation. And that's really a prerequisite to doing accurate irrigation scheduling, because if you don't have a consistent amount of water that's being applied with each irrigation, it's really hard to do irrigation scheduling. So irrigation scheduling is really a combination of deciding, when to irrigate and how much to irrigate. When to irrigate, varies a lot among growers in the early season of establishing the crop. And later in the season, when there's a lot of activity in the field, there's less choice of when to irrigate, and then it becomes more of how much to irrigate when you can get an irrigation in between spray applications or a cultivation. So, but really it's a combination of those two. And one part of this may be more important early in the season than later in the season. The approaches that are most commonly used for irrigation scheduling and deciding how much and when to irrigate can be categorized in weather-based approaches, plant-based, and soil-based. I would say plant-based is used more in tree crops and vineyards, and then in field crops, annual crops, it's more of weather-based or soil-based. Of course, maybe with grain crops, people look at canopy temperature, so that's more of a plant-based method, and it helps answer the question of when to irrigate. So this is just an example of doing accurate irrigation scheduling. What you're trying to do is really match the crop water use pattern. And so what this graph is showing is, days after planting versus the cumulative applied water to a crop. And so the green curve there is cumulatively how much water that crop needs based on what we call evapotranspiration demand. And the blue line is what we would recommend that you would apply based on, adjusting for the distribution uniformity of your irrigation system. So in the first, 10 days, we're establishing the crop, germinating up the seeds, perhaps, or establishing transplants. It has a very small canopy at that phase, but we want to get those roots growing. And so usually you have to apply more water than the crop ET. In this case, it's about 2.5 inches in the first 10 days. That's pretty good. You might have to do 3 or 3.5 inches to make sure that crop is established. Now, later on, once it's established, you still have a small canopy, and so you see the crop demand doesn't increase very much over the next 20 days. And then the canopy gets larger and the ET grows up depending on the weather, but definitely the canopy gets larger. And so that affects the crop water use. And so, of course, you would irrigate more and more as that crop increases in size and canopy. So that's the pattern we'd like to see. And you see we have to make adjustments for distribution uniformity. But by the end of the crop, we've only applied maybe 1.5 inches more than what we would expect the crop needed. So that's pretty efficient. Sometimes we see, the applied water with a curve like this type of demand curve is double or even triple the amount of water. And so in those cases, we'd really start causing a lot of leaching of nitrate. So one period of the crop where we can try to minimize those nitrate losses through leaching is really in establishing the crop, stand establishment. And over the years, we've monitored how much water growers put on, say, lettuce crops. And you see for 21 commercial fields, quite a range, depending on the weather, time of year, and the grower's practice. And then we broke this up to the water applied before the crop is thinned, so the sort of establishment water. And that averages out to about 5.5 inches. And it can be a substantial amount of the water that's applied to the crop. Here it's well over 10 inches, 12 inches. And so this is, a lot more water in many cases than the crop needed to meet its evapotranspiration demand and would cause leaching. Another consideration is how much water you apply with each irrigation. You don't want to exceed the water holding capacity of the soil. And so this table is just showing you, as you go from sort of saturated conditions to drying up the soil to say 30 kilopascals or 30 centibars of tension, for a silty clay, you're only losing about 0.4 inches of water per foot of soil depth, or a loam 1/2 inch. And if you dry it down all the way to 80 kilopascals, which is pretty dry, it's only going to maybe 0.6 or 0.7 inches of water removed. So if you apply, 2 inches of water in a single irrigation, you're going to oversaturate the soil depending on the rooting depth of that crop. So it's something to consider is the water holding capacity for these different tensions. Now, soil moisture monitoring is very popular, for deciding when to irrigate, because you can see when the soil is getting too dry. And it can be used if you're using volumetric sensors to get a sense of how much water, what the volume of water is stored in the profile. What I like about tension-based methods like tensiometers or watermark blocks is it's the signal you get, which is the tension, the matrix potential, is pretty independent of what the soil texture is. So if you get 40 centibar tension, that is an indication of how much stress the crop would be, water stress the crop would be, and whether you get 40 centibars in a sandy soil or a clay soil, it has essentially the same meaning. But one thing that I want to remark about is that soil moisture monitoring can be challenging when you have a drip system in the field. Early in the crop cycle, soil moisture is relatively uniform, and if you're monitoring soil moisture, it's really going to be helpful we're telling you you have enough moisture there that you don't need to irrigate, or you don't need to irrigate that long. So it's very useful early in the crop cycle. But later in the crop cycle, depending where your sensor is relative to the drip tape in the plant, you can get different types of information. So if you have the tensiometer or soil moisture sensor right in the crop row and the moisture doesn't move out right to your sensor, you might think it's very dry and end up over irrigating. When in fact, a lot of the roots are growing near the drip tape and intercepting the water, and the crop is actually not necessarily water stressed. If you have that sensor right near the drip tape, then you would sense that you have plenty of moisture and you might not apply a sufficient amount of water. So you really have to get out there with your shovel and your soil probe and really explore, where the water is moving around drip systems at different stages of the crop development. So I think a good strategy to figure out and answer the question how much water to apply once you decide to irrigate is to use evapotranspiration data, or we call this weather-based irrigation scheduling. So this is a good approach. I don't have time to go into a lot of detail on how to do this, but it involves using a reference ET number and a crop coefficient. And there's a lot of different ways you can find out the crop coefficient. And there's some different sources of online software that are free to use and calculators to figure this out. And as Joji mentioned, we developed Crop Manage, which can also be of assistance in figuring out your irrigation schedule based on the CIMIS reference ET data. And if you're interested, we'll be offering some workshops on how to make use of Crop Manage for irrigation management as well as nitrogen management. Now, this brings me to discussing, that another important aspect with irrigation in organic systems is when we have a sufficient amount of nitrogen in our irrigation water. And so if you're talking about well water in the Central Coast, we can have up to 70 or 80 parts per million of nitrate in that water. And we also have recycled water, which has ammonium and nitrate in it, and it can be 15 to 30 parts per million in that recycled water. So we want to definitely take credit for this nitrogen that's in our irrigation water when we're doing our planning and doing our fertilizer recommendations. So in the past, we did replicated trials in drip in lettuce to simulate waters with different nitrate concentrations. And we had from 2 to 42 parts per million nitrate in our different irrigation water. And we just irrigated the crop with these waters, no fertilizer at all, to see and prove that this nitrogen in the water has value. And so you can see, it affected both the plant size and the end content of the tissue and the recovery of the nitrogen. And it worked just as well as using fertilizer. And we did this in conventional systems, but it's the same concept for organic systems. And if you want to learn, more about those trials, encourage you to go online and there's an article published in California Agriculture. But the calculation for taking credit for nitrogen irrigation water is very simple. It's the applied water in inches times the nitrate N concentration of your water times this factor of 0.23. And that gives you the pounds of N per acre that are applied with that water application. And so for an example, if you're applying 2 inches of water and your nitrate N concentration is 30 parts per million, It's 2 inches times 30 parts per million nitrate N times 0.23. That's about 13.8 pounds of N per acre that you're applying with those 2 inches of water. And if you were thinking about 8 inches of water being applied, that would come out to be a significant amount of nitrogen. And so this table just shows you for different nitrate concentrations, the pounds of N per acre foot of water applied that would go to your crop that you could take credit for. So it can be very small amounts if it's 5 parts per million, but once you get up into 40, 50 parts per million per acre foot, you might be contributing 140 pounds, 150 pounds of nitrogen per acre foot of water applied. So very significant amounts. But there's some, practical challenges to taking credit for this nitrogen in your irrigation water. It's the case on at least on the Central Coast, and maybe the case, in the Central Valley. You may have multiple water sources that you're using to irrigate your crops. You might have 2 or 3 wells on your ranch, or sometimes you use surface water, sometimes you use well water. And so it becomes a little harder to make that calculation. Some of our wells, their nitrate concentration changes throughout the season. So there's some monitoring that you'd need to do. And then you need to know how much water you're applying, whether it's with a flow meter or you have a good sense of how much water you're applying with each irrigation. Another question that growers have is how much credit should they take for nitrogen in the water during pre-irrigation and germination? And the most practical thought is that the applied amount of water is much greater than crop evapotranspiration in this period, especially when you're doing pre-irrigation and you've just done your first irrigations to sort of wet up the soil to germinate the seed. And crop N uptake, of course, would be very minimal. So this is not really a period to take credit for the nitrogen in your irrigation water. It's after the crop is established would make the most sense, when crop evapotranspiration is actually increasing. So that's an important consideration. But this is a period not to over irrigate, of course, and leach that nitrate out. Some other practical considerations in taking credit for N in the water is that if you're thinking about, what would be the right amount to credit, at a minimum, you could take credit for the crop ET volume in the irrigation water over the season. So if it's 8 inches of ET, you could take credit for 8 inches of water that's being applied, assuming you apply at least 8 inches and the nitrate in that water. Something else that we've seen over the years is that even though you're applying fairly high nitrate water, unless it's really high nitrate water, you may not see any impact on soil nitrate levels. It's really sort of a leap of faith that nitrate in the water is going into the crop. It's not like you apply the water and then you suddenly see your soil nitrogen levels jump up on you. You're hopefully applying that water at a rate that the crop needs, and so you're meeting the crop demand that way. One of the nice things about this is if you're irrigating really close to the crop water needs, the crop water needs are very much in sync with the nitrogen demand of the crop too. So that's one reason the nitrate in the water can be a very efficient way of getting nitrogen into the crop, because the ET goes up just when the crop end demand also is going up. So they're really in sync. And so if you really do good irrigation scheduling, you'll make very good use of that nitrogen in your irrigation water. And then this is a practical way you can evaluate the nitrate content of your water if you're blending different sources, and simply using a emitter on your drip system, or you can even do this on a sprinkler system and putting the water in a bucket. And then using that soil nitrate test to evaluate the water that you collect periodically. And if you're using different sources, you can get a sort of a blended measure of the nitrate concentration of your water. Now, in a practical sense, you might ask, well, how do I manage looking at my soil nitrate and making decisions about fertilizing and factor in the nitrate in the irrigation water? And the way I think of it is, if we look at our soil nitrate concentration at any point in time, you're looking at the current nitrogen status of the soil. If you're looking at the nitrate in your irrigation water, this is a future contribution of nitrogen when you irrigate. And so you have to blend those two ideas together. And so we put that strategy to a test, running some field trials where we had some strip treatments with the grower practice, best managed practice, where we use this concept in a sort of intermediate practice where we make either higher or lower applications of nitrogen depending on where the grower is in this, in his strategy. And just to quickly show you, here's 5 trials, and you see we have different soil nitrogen levels at the start, and this is after side dressing. And we have variation in our nitrate in our irrigation water from 32 parts per million N all the way up to 84. And then you see the amounts of water we applied after thinning. And so here's our low and our high treatments, and then trial 5 is sort of mid. And then this is the amount of nitrogen that was applied through that drip irrigation. And then we did have variations in the salinity of the water too, so we wanted to monitor that. But then we did, our practices in nitrogen management, looking at the nitrate concentration of the water and looking at the soil nitrogen, and we did yield evaluations. And compared to the grower's yield, we had similar or even higher yields than the grower treatment, using less nitrogen fertilizer. And this is just a summary for all the trials of the nitrate concentration in the irrigation water in parts per million N versus what we saved in fertilizer, using 175 pounds of N as a standard, which is the average amount that growers are currently using for lettuce. And you can see when we get up to 60, 70 parts per million N, we can save 100, 140 pounds of nitrogen fertilizer. Now, these trials were done in conventional, but the same concept really applies to organic systems. So I'll just wrap up, by just summarizing that really the key in water management strategies for efficient use of nitrogen in organic systems is to ensure that your irrigation system has a high application or distribution uniformity, then match the irrigation schedule with the crop water requirement. So really look at ET. You want to minimize applying too much water for stand establishment, and you don't want to exceed the water holding capacity of the soil when you irrigate. And then finally, you really want to take credit for the nitrogen in your irrigation water. So with that, I'd be happy to answer any questions.
Thank you very much, Mike. Yeah, time is not much left, but there's one question. So here we go. What are the best resources for crop coefficient other than crop manage? What is the coefficient that are used by crop manage?
Yeah, so you don't have to use crop manage. There's other online calculators that you can use, but there's some published crop coefficients. You can look up on some of our drought. There's UCNR publications called Drought Notes, I think, and they talk about how to make these calculations. You can do it all by hand, and you can base that on canopy cover. So, those are some of the tools that could be used. But the issue is for vegetables, there's no one number for a crop coefficient. It's always changing through the season.
Basically, you have to measure canopy cover throughout the growth season.
Yeah, and then convert that to a crop coefficient.
Okay. Okay. Well, thank you very much, Mike.
Yeah.
Appreciate it. All right, let's move on to the second speaker, Margaret Lloyd. She gave a talk last week, excellent talk on cover crops. So you need not much introduction, but she's the small farms advisor for Yolo, Solano, and Sacramento counties. Okay, Margaret, take it away.
Great, thanks, Joji. What a great talk, Mike. Thank you so much. Okay, so today I'll be talking about estimating N availability in compost. And so composting is achieved in a variety of manners, as many of you know. So from static aerated piles to windrows to top-fed vermicompost bins. And in this process, most of the decomposable or mineralizable forms of nitrogen are converted to more stable forms or lost as ammonia gas. And so it reduces the mineral N content and increases the stability of the organic matter. One way to study the compost N mineralization is through soil incubations. And to do this, the compost is added to field soil. And in this case, with the work that we did, we added it to organically managed field soil from local farms and incubated it in optimal temperatures and moisture for 12 weeks. And Patricia will describe this a little bit in more detail in her next presentation. But so what we learned from this process, we took multiple types of compost, those based from poultry manure to yard trimmings to mixes of the two of those. We had some vermicompost. And this is what we found. We found that the composted, we have simplified here, the composted poultry manure had some readily available nitrogen at the beginning. So if you look at time zero on the bottom x-axis, you have the time, and on the vertical axis, you have the available N. So you can see that that composted poultry manure had some initial nitrogen available. And you sort of see these two phases looking at that composted poultry manure. And you'll see this with amendments that Patricia will talk about next, the sort of initial, somewhat rapid release or available release, that sort of steep curve, and then a second, more slow release rate. And so when you look at the end of 12 weeks, so that would be, about 3 months, you can see that somewhere around 30–35% of the total nitrogen that was in that material became plant available. And when we look at the composted yard trimmings, it's hovering around zero. So there was little to no to even tied up nitrogen from that type of material. And composts are sometimes applied to supplement the soil nitrogen. However, if that is really the goal, the nitrogen availability from these materials differs widely. And so here's just a table form of what we just looked at with a little bit more information. We present these two types of composts, a yard trimmings based and a manure based. And you can see what the typical percent nitrogen is from less than 2 to more than 2, what that C to N ratio looks like. So the yard trimmings was in the range of 13 to 20, and the manure based compost was 6 to 8. And then that nitrogen availability after 12 weeks. So thinking about from a nitrogen standpoint, the yard trimmings compost is going to be providing nitrogen in a long-term sort of years time frame, whereas the poultry manure compost is giving some of the nitrogen in the near term, in the weeks to months time frame. And what we found was that the carbon to nitrogen ratio of an organic substance, and especially its degradable fraction, is the decisive factor for N release from organic matter. So this was a really important predictive factor for how to estimate what you might get from a material. So here is a figure. On the x-axis on the bottom here, you can see the carbon to nitrogen ratio. And on the vertical axis, we have the available nitrogen across 12 weeks, at 12 weeks. And that's percent of the total. And so what you can see here, so the two composts are these red dots and blue dots. And you can ignore the other dots for now. We're going to look at those during Patricia's talk. But so the red dots are here, and the blue dots come right across the zero line. And you can see that this curve crosses somewhere between 15 to 1 and 20 to 1. So this becomes an important threshold to make note of for where N immobilization could be occurring with a compost. So this means that it's not going to be a source of nitrogen, and it can even begin to tie up nitrogen from your crops. In the absence of a C to N ratio, the N concentration can be used. So this is the N concentration on the x-axis instead of the C to N ratio. And so if you recall, the plant-based compost was like 0 to 2% nitrogen, and the manure compost was in the 2 to 5 range. So you can see how they also follow a correlate well to a curve here, looking at availability by percent nitrogen. That being said, most compost suppliers are able to provide you with a C to N ratio. So on that note, a compost supplier will usually be able to share with you a soil test or a compost analysis. And this is what it might look like. So just to point out a few key points here. So in this analysis, this is a part manure, part yard trimmings-based compost. And we have percent. So this is about 2% nitrogen. And note whether the report that you're getting is reported on a dry basis. If not, the calculation for moisture will need to be taken into consideration. Also, you'll commonly find this C to N ratio here. So with those pieces of information, you can, that's what they've done here, is this calculation for pounds of nutrients per ton. So what they did was take the 2.09%, divided by 100, and times 1 ton of material, 2,000 pounds, and that gives this 41.8 pounds of nitrogen per ton. So that's the total amount of nitrogen in the material. How do we estimate plant available nitrogen? So I'm going to use these two figures that I showed earlier and walk through that. So we have a part manure, part yard trimmings-based compost. And while this figure is simplified, we generated this figure from multiple types of compost, multiple batches of the same type of compost, and we found that it was quite consistent, and that if a material was roughly half manure-based, half yard trimmings-based, it really fell right in the middle here. So that's what we're going to do for this part manure, part yard trimmings compost. So we did this first calculation. We got the total amount of nitrogen in 1 ton of the product. So the next step is availability. That report showed 11 to 1. And this falls about right in the middle. And so just visually, I'm going to estimate that at around 15% of the total. You can also look at this table. 11 to 1 is about in the middle between these two. So I took about half of the poultry manure compost. multiplied the total times 15% of the total to get about 6 pounds of nitrogen per ton material. So this is what can be expected in the following few months after application. Of course, this has to be calculated by the amount applied. So I chose 10 tons per acre. So that gives me about 63 pounds of nitrogen per acre I can estimate will be plant available from this compost. One note is whether the material you bought was calculated by, was it tons per wet weight or tons per dry weight? So moisture can range from 30 to 60% of material. And so considering the actual application and making adjustments for the amount of nitrogen by dry weight is always the easiest. So here's another soil test that we can look at together. This is for spent mushroom compost. So the total nitrogen was 1.75%, so just under 2%. And they also have in this soil test ammonia nitrogen, and that was at 1.22 pounds. The C to N ratio is 22 to 1, so that's quite high, and we might expect some tie-up, and 58% moisture here. And in this material, they also provided bulk density. So if you're working on a cubic yard calculations, this can help you make that conversion from tons to cubic yards to tons, so that you can work in the pounds per acre, tons per acre conversion. And the reason that I bring up the ammonia is because that could be suggestive of how much nitrogen might be available immediately. So if you recall that the poultry manure did have some more immediate nitrogen available, that can be interpreted somewhat from having a number like this on a soil test. Now, this is a very small amount in this case, so I would not be expecting much from this compost in terms of nitrogen. So one comment is that high mineral nitrogen contents usually lead to good short-term and availability. So following up what I sort of just talked about, and here's that starting point for this composted poultry manure. So looking for that piece of data in a soil test can help you refine what you might expect in the short term. So if only a small part becomes available each year, how much is ultimately available? And what's that N accounting over time? This is a really beautiful visualization of that cumulative impact of annual composting. So let's follow the blue color. So each of these colors represents a compost addition. So year one, we add compost, and we can expect about 33% of that total N, the N mineralization, from that compost that year. The second year, that material remains in the soil and is going to be releasing an estimated 16%. The third year, an additional 9%. And in this way, you can see how this material is providing a sort of background contribution of nitrogen to the system. Because in the second year, then, you're going to add another 43 pounds of nitrogen per acre from a manure-based compost. And then you add another 43 pounds of nitrogen per acre of compost. And so by adding it each year, you can see that there's this cumulative effect of nitrogen benefit and release. And after about 4 years, you can see it sort of reaches a steady state. And some of you, I feel like I have often heard about when folks are transitioning from conventional to organic farming, there's this transition period. And there's a lot of things biologically that are getting sorted out during that time. And I feel like this visualization is a way to understand some of what might be happening with the nitrogen cycle in the soil during those early years of a transition, or even just starting to use compost. So the full benefits take several years to accumulate and start to reap the full benefits. And if you recall this visualization, this figure from last time, in which we talked about all the contributions of nitrogen from multiple inputs. And so this blue band represents the compost addition. And you can see here that it's about 40 pounds or so we're expecting from this compost composted manure. And so that what I just showed about the accumulated effects over time, that's coming from the soil organic matter. And so in a way, when we think about the soil organic matter band, it might be like a rainbow of bands, because it's releasing nitrogen from all these different things that have been happening over many years. And one of those being the repeated application of compost over time. And so after the long-term application of compost, the overall end use efficiency, when it's compared to a mineral fertilizer equivalent, is typically reported in the range of 40 to 70%. And so that's thinking about the bioavailability. And it's often assumed that the mineral fertilizers are usually at 100% availability. Another way to think about this and look at this is in these sort of active pools and old pools of soil organic matter. And so when we look at the age of residue, that can be crop residue, compost, we'll see that in those first years following incorporation, they can be considered in this active pool of soil organic matter or of organic matter. This is going to be our crop residues, manures, compost, cover crops, and they're going to be continually releasing and breaking down, similar to that stacked bar chart showing the compost over time. So you have these active pools for the first 4 or so years, and then they become these very stable forms of organic matter that are very slow to break down. And I could end this talk here because this really covers the basics and the drivers and the nuance of some of the availability. Because once we go on beyond that, it's kind of like opening Pandora's box. The variability in feedstocks, compost age, compost maturity, moisture, compost process, they all become variables that influence this wide variability that many of you might be familiar with compost. So that being said, I will cover a couple more points, but I really want to emphasize that you can go a long way with understanding what to expect from a compost with just knowing the C to N ratio, and then some of the additional information can help with some of the detailed nuance. So what makes compost different from crop residue and even other types of amendments? The main difference is that during composting, the easily decomposable or labile materials are eliminated, and what is left is a more stable form of carbon present in the compost when it's applied. And so materials such as food waste, biosolids, or manures are rich in fats, sugars, starches, and waxes. And these labile forms are quickly converted by compost microbes to CO2 and water, and they leave behind recalcitrant resistant materials such as wood And these tend to contain a lot of cellulose and lignin. And it's common for these initial labile materials to generate odors, to tie up nitrogen, to release phytotoxic compounds as they break down. And because of all this activity associated with the biological breakdown, the most direct means of assessing a compost stability is to evaluate the rate at which CO2 is produced as the material breaks down under controlled conditions. So this resulting material, one of them is lignin, and lignin is very tough to decompose. And I have this image here to show how complex lignin is. And it's the second most abundant component of plant residue in terrestrial ecosystems. And by itself, it's relatively recalcitrant. And degradation rates are negatively correlated to the lignin content, or the lignin to N ratio. The purpose of lignin is it serves as this cement between wood fibers, as a stiffening agent within the fibers, and as a barrier to enzymatic degradation of the cell wall. And so one tangible way to identify lignin in your compost is how tough that material is. Can you tear it? And, lignin forms a physical barrier by attack from enzymes, and it reduces the accessible surface area of cellulose. And so it's very slow to decompose. And in addition, in order for it to decompose, it often has to have a co-substrate, carbohydrates. And so there's multiple reasons why lignin can be hard to decompose. And when microbes do decompose it, they basically break it into these smaller molecules, complexes. And so one reason why some of the high nitrogen products may have lower availabilities is the amount of lignin that is in that material. Now, this is still often embedded in the C to N ratio, but it can be an added factor for part of the slow release of a material. And so the higher the lignin fraction, the less mineralization will occur. Now, here's an example. If we compare alfalfa pellets, which have a pretty high lignin to N ratio, compared to partially composted manure. And so these alfalfa pellets are going to have less N availability relative to this partially composted manure. And if we look at blood meal, it doesn't contain any lignin. So a couple more thoughts. Composting plant residues increases that lignin content, and it decreases the sugar and lipid contents. And in this study, what they found was that the labile portion of the plant residue was significantly decreased from 30% to 9%. And what this is doing is this is decreasing the energetic compounds for the microbes, the sugars, et cetera. And so there's less available for the microbial food. Composting plant residues increases by threefold their residence time in the soil. So that material is going to stick around longer and be part of that active but slower pool of release over time. And lastly, the addition of fresh plant residues causes a priming effect, but compost residue does not. And that priming effect is that initial sort of flush of activity when material is added. It's a stimulation of the native soil organic matter degradation. And so what you can see here, this is total CO2 evolved. So this is somewhat representing like the microbial activity when material is added. And so here, this is non-composted plant residue. The darker circles are composted plant residue, and this is the control, the baseline. So you do see an increase, but not as much of an increase comparing compost to plant residue. And this is another visualization showing plant residue mineralization, the percent of the initial. So you can see this is less than 40% of the initial amount was mineralized compared to the non-composted plant residue. And the last point is that the amount of liquid contained in the compost affects the concentration of nitrogen per ton. And that mineralization of these organic materials is mediated by bacteria and fungi. And so the soil moisture will regulate that oxygen diffusion in the soil, with the maximum aerobic microbial activity occurring between 50 and 70% of water holding capacity. So moisture will influence how much one can expect from their compost. So to summarize, these two figures, whether you like to read it as a table or see the curve, are very useful tools for just that baseline understanding of what's the important C to N ratio or percent nitrogen of a material that can help you predict what you might expect for the nitrogen contribution. And 15 to 20 is that threshold for N tie-up. Second, that high mineral nitrogen contents usually lead to good short-term N availability. Third, the lignin to nitrogen ratio is negatively correlated with N mineralization rate. And that there's a big difference between adding fresh plant residue, which causes that priming effect, but composted residue does not. So thank you for your interest and attention in compost and nitrogen mineralization. And I'll take any questions.
Thank you very much, Margaret. Excellent talk. All right, so there's one question on the Q&A board. By adding yard trimming would have the ability to hold on to nitrogen in soil, so one does not lose nitrogen to leaching would cover. So one does not lose nitrogen to leaching would cover crop hold onto nitrogen as well.
Yeah, so that's such a somewhat revolutionary idea of how to think about compost, less so as an amendment that is contributing nitrogen to your soil, but rather as a tool for capturing soil available nitrogen. And I think it's the folks on the Central Coast that really opened up my mind to that possibility. So absolutely, the high carbon, high C to N ratio composts have that potential to capture soil nitrogen, residual soil nitrogen. And I think probably Joji and Richard can comment more about maybe some of the work that they're doing on that. And would cover crops hold on to nitrogen as well? So we did talk about the grasses having these really deep, fibrous, efficient root systems that can sort of mop up available residual soil nitrogen. So the grasses in particular can be really good at that. And legumes can do that too. Legumes will preferentially use residual soil nitrogen over making nitrogen in the nodules. So they are able to do that too, but just do it to a lesser extent than the grasses, or the root system is less complex and doesn't cover as much soil area as the legumes. I mean, the grasses cover more soil area than the legumes. So I hope that answered your question.
Yeah. The grasses goes deeper too, huh?
And deeper, yes, exactly.
Yeah. Okay, any other questions? You can type it in the Q&A. Oh yeah, here's another one. Okay, yeah, green waste compost seems more strategy to increase soil carbon, and organic matter percent for long-term nutrient cycling improvement. But we need to be supplemented with additional fertilizer inputs, probably for years, to meet crop demand, crop needs, until such time as the organic matter level is sufficient to support nutrient cycling. And the cover cropping and compost application are complementary practices, not similar alternatives for organic nutrient management. Is it best practice to apply compost when cover is incorporated in that case?
Yeah. So maybe, I wonder if you're thinking about that similar topic to what we just spoke about, which is trying to capture some of the nitrogen from the cover crop release. Since you're asking specifically about green waste compost, which would be the equivalent of what I was calling yard trimmings compost. So that's an interesting idea. My initial thought is that could be used in that way. Yeah, but maybe others have some more thoughts on that too. Yeah. Mm-hmm.
Okay.
I mean, sometimes, a lot of times it can be hard to make choices like the order of operations. So, when you add compost, I think, logistically it can be done after cover crops. So
Right.
Seems like an option.
Okay. Any other questions? All right, coming more here. But because compost doesn't have labile carbon and doesn't have the priming effect, would it diminish the nitrogen immobilization potential?
So the immobilization potential will have to do with how much carbon is in the soil. So, I mean, I suppose, it's hard to make a generalization because there will always be these types of carbons going into the material, into the soil. But certainly, as that carbon gets worked through by the microbes, the issue of immobilization will be reduced, if that makes sense.
Okay.
But, I think it's also important to continue to recognize that in a way, N availability is a byproduct of microbes utilizing their energy sources of C, of carbon. So in order for a lot of this mineralization to occur, microbes to have these flushes in populations and turnover, it's really a response to their access to labile carbon sources that are stimulating all of their activity. And as a byproduct, we often will get this nitrogen availability. So they are linked still. But the quality of C will be influencing how much microbial activity we might expect.
Yeah, as just Margaret mentioned, the quality of C is kind of important. Margaret mentioned the lignin content. Lignin is a type of carbon compound in the soil. And then C/N ratio is just talk about the total carbon amount and total nitrogen amount. It doesn't consider quality of carbon. So that's another factor that affects mineralization or immobilization. Okay. Another question here. Do you see nitrate numbers in the soil go up or correspond with irrigation with the high nitrogen content irrigation water? Oh, this is a question for Mike, I guess. Mike?
Yeah. Hi, Joji. Yeah, I was talking about this at one point in the presentation, that we don't see that the soil nitrate numbers necessarily go up with the high nitrate water. What can happen is in places where there is high nitrate water and growers aren't taking consideration of that high nitrate water, they over fertilize essentially. And then those sites tend to be sites or fields that have high soil nitrogen, just because there's so much N being applied. But we've had other fields, you know, where soil nitrate numbers are actually quite low, even though you know, we add high nitrate water. In the trials we did at Spence, for example, we saw no correlation between the nitrate in the soil and the nitrate of the water that we're applying to those different treatments. And that was from 2 to 42 parts per million N.
So it depends, huh?
Yeah. So it's really, to me, it wasn't that intuitive, but I guess, you know, if you're applying the water through a drip system and you're pretty much keeping up with the demand of the crop, that nitrogen in that water goes into the crop and it doesn't hang out in the soil.
Right. Yeah, that's a good thing. That's very efficient.
Yeah, it is. That's what it taught me, is it doesn't just accumulate in the soil. And remember, nitrate doesn't stick to soil, it moves. So if you over irrigate, what are you doing? You're leaching some of the nitrogen actually that's already in the pool. And so you may not see that nitrate increase. It's not like you just keep adding water and the nitrate sticks and the water goes away. It goes with the water.
Great.
Yeah.
Okay, probably one more question, last one, to Margaret. Does lignin eventually break down over time, or is it constant?
I'm not sure how long. This is somewhat going beyond my depth of knowledge. I mean, it can stay around for a long time. You know, after those initial years, I think it sticks around for a very long time. Maybe others have some thoughts on it.
Yeah, I think it's a very long time, years. It's very difficult to decompose biologically. Yeah.
Mm-hmm. Yeah, I mean,
like,
a piece of wood is a good example of, you know, you can
yeah,
maybe have trapped that in your own yard, how long it takes to break down.
Like a bark of redwood bark.
Mm-hmm.
Yeah. Okay, well, thank you very much, Margaret. Appreciate your talk.
Thank you.
All right. So the final speaker for today is Patricia Lazicki, and she's going to talk about estimating nitrogen from organic fertilizers. Brief introduction, Patricia is the PhD candidate in soil science at UC Davis. Actually, she's going to give an exit talk on Friday, so almost done. Previously, as a soil science researcher, she worked to develop the nitrogen guidelines for crops throughout California. And her research continues to focus on soil health and fertility in organic cropping systems. All right, Patricia, go ahead.
Thanks very much. So my part of the talk is going to cover fertilizers. So what makes fertilizers different than some of the other things that we've been talking about? First off, to be sold as a fertilizer, it has to have a guaranteed minimum nutrient content. And they're also often processed in some way, maybe chemically, maybe physically, but usually in such a way as to make the nutrients more readily available. And so they'll be more quickly available, less dependent on some of these other factors. They also tend to have less variable composition, so they can be applied more uniformly. And because of these processing steps, they're also tending to be much more expensive. So these are things you're using more in smaller quantities when you want to some targeted application. So just brief recap of some of the things that we've been mentioning over and over again. When you apply nitrogen in an organic form, it's in more complex molecules, the plants aren't going to be taking them up. So bonds need to be broken by enzymes in a gradual process. We think of just as generally breakdown into simpler and simpler molecules. When mineralization happens, that's when that amino group is broken off. You get ammonium, which is a form that plants can take up, and then that very quickly under warm and moist conditions is converted to nitrate. So nitrate's going to be the form that's most quickly available to plants. As Mike was mentioning, it's negatively charged. The soil surfaces are also negatively charged. So it's not attracted to soil surfaces. It's moving with the water, very quickly available to plants, very quickly lost. Whereas ammonium, that has a positive charge. It's going to be attracted to soil surfaces. It'll stick around in the soil longer, but also be a little bit harder for plants to get off the soil surfaces. So with a lot of the pre-treatments for fertilizers, part of the goal is to accomplish this preliminary breakdown step. And because all of these processes are done by enzymes, they happen faster at higher moisture and temperature. But because with fertilizers, you've already done some of this breakdown, they're going to tend to be a little bit less dependent on those factors. So in this presentation, I'm just going to go through a couple of different types of fertilizers and talk about their different behavior. So we'll be going through slaughter products like the blood and feather meals, is different patented granular formulations that combine a lot of different sources, and liquids. So a lot of these numbers come from this experiment which Margaret referred to, where we combined different amendments with soil in the lab. We incubated them at optimum moisture and temperature, so similar to an irrigated agricultural soil during summer. And then we took samples right at the beginning to get that initial available nitrogen content, and then periodically over 12 weeks until they formed kind of a plateau as an index for what's going to be available during the season. And then for a couple of amendments, we were interested in looking at the difference that incubating at different temperatures made. So we incubated a couple at colder temperatures. So I'm going to start with slaughter products. So these are blood, fish, feather meals, bone meal. As Margaret was mentioning, a lot of these, they're animal products. They don't have lignin or this very hard to break down stuff. And a lot of them are mostly protein. So as you can see, looking at the label here, very high nitrogen analysis, often have low phosphorus and potassium. A big exception to this is the bone meals, which have a lower nitrogen and high in phosphorus and calcium. And because they're mostly protein, they tend to have very low mineral N in the initial product that you apply, but it's really quick to break down in soil. It's really quickly released. And some of these can also be soluble, so they can be, they're marketed to be fertigated. So this is the results that we found in our incubation. On the y-axis here, you see the amount of the nitrogen, or the percent of the nitrogen that we applied that was available over the course of time. So as you can see, very little mineral nitrogen at the beginning, but extremely rapid mineralization within that first week. And pretty much everything that it was going to release, it had released by 6 weeks after we incorporated it. And pretty uniformly, everything tended to release around 60 to 70%. And this is something that, as you look across the literature, is a very constant number for these slaughter products, at around 60 to 70%. Granular products are usually, they're patented blends that include a lot of different kinds of things. And so the particular characteristics of the fertilizer will depend on what went into it, how it was treated, whether it was ground or pelleted, heat treated or whatever. So just taking a look at this label here, this particular 442 was derived from chicken manure, raw fish, elemental sulfur. So chicken manure, it has a lot of ammonium in it initially, and that's what we found for a lot of these products that were partly derived from manure. So if you look at this label here, 4%, it's 4% nitrogen, and of that, about 10% of that is already in the form of ammonium. So one of the implications of that is if you apply it on the surface, ammonium that's applied to the surface is easily volatilized, especially if the soil is kind of alkaline. So that's going to be important. And then also that's nitrogen that's also immediately available to the plants. And we're also including guano in this category. This is a pelleted seabird or sometimes bat manure. And this also tends to be very high in nitrogen and in phosphorus and very readily available. So here's the results from our incubation. As I was mentioning with the guano, this isn't necessarily typical, this result where it started with over half of its nitrogen already in mineral form, but they do tend to release everything almost right away. So you can see really even most of what was available after the first week or so, and almost 100% by the end. With some of these different pelleted formulations, there was a bit of a range in the initial ammonium, and again, depending on what they're formulated from, but the amount that was available by the end pretty much tended to follow the guaranteed percent nitrogen. For the liquids, these are often fish, plant, guano-based, and they're designed to be immediately available. So often pre-treated to increase availability. So if you see hydrolyzed, that means that it's been pre-treated with enzymes. Emulsion, that means it's been heated to accomplish some of the same things, breaking down those more complex organic molecules. An important thing with liquids is we've been talking about looking at the percent nitrogen, is that when they're marketed, the percent nitrogen is of the fresh weight. And so you'll notice that a lot of the other materials that are kind of the high availability are also high nitrogen content, like 12%, 13, 15 for some of these guanos and slaughter products versus this 4%, but that's because it takes the liquid into account. It says a percent of the wet product. So in our incubation, we found that these followed a very similar pattern to the blood and feather meals with their availability. You were applying a fair amount of mineral nitrogen initially with these, a bit more than with the slaughter products, but very rapid increase within the first week and kind of a slow release after 3 weeks. One thing that I want to note with our particular incubation is that we used a uniform rate for all of our amendments. And so we applied at what I think is a pretty high rate for these. So you can see in the literature numbers of up to 90%, 100% availability. And we didn't test it, but I suspect that partly the reason why the availability wasn't as high is that we were applying at a very, very high rate, and that you probably get a greater availability when you apply at a more normal rate. So you'll see in the materials that we give a range from about 70 to 100% availability for these. So just as a summary for our findings, the this table ranges from like a lower percent to a higher percent, and lower percent or lower availability to higher availability. So your granular fertilizers, of course, depends on what they're made of and how they're treated, about 2 to 7%, and tended to have a C to N ratio of about 5 to 7. About 40 to 60% of the nitrogen you could count on becoming available, and then the release within days to weeks. With these other higher availability materials, much higher nitrogen percent, except of course for the liquids, because they're on a liquid basis. C to N ratios much lower, and then availability from 50 to 100% depending on the material, and then very quickly available, usually the greatest release within the first week. So it's kind of a good place to start in predicting whether it's going to be available or not, is looking at the chemistry. The temperature and the placement are also going to make a big difference in estimating the availability from fertilizer. So we're going to go through a couple of how you make that calculation. So this is the same graph you've seen a couple of times with Margaret's presentation, and just showing these top dots, the green and the blues and the purples. These are the fertilizers, and the potential availability was pretty well related to the C to N ratio, so can make a broad guess based on that. It also had a pretty good relationship to percent nitrogen, except of course for the liquids, because they're on a fresh weight basis. So the C to N ratio was a pretty good place to start for these. Here's some data. This is both from our lab and from a couple of studies by Tim Hartz, looking at what effect different temperatures have. So we've mentioned a few times, nitrogen release is an enzymatic process that's slowed by colder temperatures. So when we look at these graphs, this is again the available N is the percent of the applied N, and how it's released over time at different temperatures. So this is from our lab, and we tested a potentially like cool winter temperature, 40 degrees versus warm summer temperature, and we did see slower availability, but not actually that much delay. So looking across the our granular fertilizer, our feather meal, you reduced it, but not by more than a couple weeks probably. There was a tendency for total available nitrogen to be slightly lower at colder temperatures, on average about 10 to 15% when the temperature was kept consistently low. This is some data from Tim Hartz where they looked at the effect of different temperatures on liquid fertilizers and found that it had very little effect, which makes sense given that liquid fertilizers are pre-treated to make them almost immediately available. So those things which are affecting enzymatic reactions will have less effect on those. Something that these graphs don't show, because they show only the total mineral nitrogen, is that colder temperatures really do delay nitrification. So again, most of the nitrogen stays in the form of ammonium for longer than they would in warmer temperatures. So again, this means it has less risk of leaching, but it also may take a bit more time for plants to get at it. And then finally, I want to go over the important issue of placement. And so this is some data from an experiment that Richard Smith did. So if you have questions about it, I will refer them to him, but take some 442, put it in buried bags, or either buried in the soil or placed on top of the soil, and then measure the mineralization over time. And it made a big difference. This black line is the buried bags. They're in better contact with the soil, in better contact with the microbes, they're more moist. And so it ended up being about a 30 to 50% reduction in the amount that was available. And then of course, when you place it on the surface, if it has a high ammonium, then that's also more risk of volatilization. So just as a summary, in predicting your nitrogen availability from fertilizers, first you can use the chemistry to get an estimate. So based on the percent nitrogen or the C to N ratio, and then taking into account the temperature, reduce it slightly if you're going to be applying more complex materials in cold weather. And then if the placement's going to be on the top of the soil, reduce by about 30 to 50%, and reduce it by a bit more if you know that the initial ammonium is high, or if it's going to be in dry conditions, because then it won't get washed down into the soil as much. And then wanted to end with a bit of a note about timing. So this is a version of the same graph that you've seen a couple of times. This is a modeling exercise that we did using temperatures and amendments in Yolo County. And then looking at this graph shows the daily release from different amendments versus the daily uptake rates of tomato. So what I'm wanting to show in this graph is just when you add, this is a granular fertilizer, 444, using the parameters that we determined from our incubation, you get a big pulse of available nitrogen right away. And then this available nitrogen is hanging out in the soil until it's taken up by the crop or washed down and leached by irrigation until it's taken up by the crop. In comparison with, this is the same exercise done as a side dress. So in case A, we applied it right at planting. In case B, we applied it at early flowering. So again, you can see there's that quick pulse of available nitrogen. It mineralizes pretty rapidly, and most of it is available right when the crop is starting to do rapid uptake. And again, here we modeled adding a water run application of a liquid fertilizer. So again, you can see the potential benefits of having something that's rapidly available when the crop uptake is rapid. So I think that this goes along well with Margaret's presentation about composts, because composts are going to be building the soil fertility over time. So that's going to be your basis and probably most of the nitrogen that you're applying, but then fertilizers can be very helpful for quick targeted applications when the crop needs it. So I'd like to, thanks very much for your attention. We actually published some of this data in the Journal of Environmental Quality. So if you're interested to get more information or see some of the numbers, it's available here, and happy to take questions.
Thank you very much, Patricia. It was very informative. Okay, one question on the board. Any thought on plant-based fertilizer, for example, soy meal? Is there any nitrogen, any more plant available, or otherwise more quickly acting? Any other consideration with plant-based fertilizers?
Mm-hmm. Yeah, a lot of it we found was really well predicted by the percent nitrogen. So with our tests, at least with the ones that we tested with, and with some of the ones that I've looked at in the literature, it's predicted pretty well by the percent nitrogen and by the C to N ratio, sort of despite where it comes from. So I know that Tim Hartz's lab did some work with hydrolyzed soil or soy, trying to remember if it was a liquid or not, and tended to have a bit less availability than some of the guano-based, but kind of following a similar pattern of release and broadly predicted by the percent nitrogen.
Yeah, I have experienced using one of those material like hydrolyzed soy protein. Yeah, it's soluble in water and quick really fast. Release nitrogen pretty fast. I haven't done any mineralization experiment, but I'm going to do it. So yeah, maybe sometime soon I have some data to share.
Nice.
All right, any other questions here? Oh, just one more. The incubation experiments, did you grind or otherwise prepare the granular blends prior to using them?
Not much. We did it in really big, we actually did it in red solo cups. We could use a lot of soil. So we had to only grind the mineral, or with some of them, we had to break them apart a bit, but we didn't grind them much. But yeah, that's a really good point, is that the size of the pellet is going to make a difference in how it releases and how it spreads.
So you didn't grind, used that as is?
Yeah, we used them as is. With the guano pellets, we broke them up a little bit, like just took a mortar and pestle and just kind of crack a pellet so it could to break it up a bit, but otherwise not really.
Okay, thank you. All right. Well, any question, not just Patricia, but you know, today's all speakers, and also Daniel's here too. So, you know, even question for the last week speakers is welcome. Any questions? We still have plenty of time, 15 minutes or so. So please feel free to type it up, your question. Here we go. All right, another question. Do you think that the mineralization rates in the lab incubation would have differed if fresh plant residue had been added?
I think I guess the answer is always it depends a bit. With fresh residues, the quality of the initial carbon makes a lot more difference than in either composted or fertilizer materials. So it really depends. Like the literature that I'm familiar with compares fresh manures to composted. And there you quite often see, even with a pretty high, a pretty low C to N ratio, you often see a quick initial immobilization because the microbes are excited about all that fresh, delicious carbon. And so quite often, a lot of the nitrogen that you add initially is immediately immobilized and then slowly released. So that's the literature that I'm most familiar with, and just you see a lot of variability because there's a lot of variability in the quality of the initial carbon. For our compost that we added, we noticed that of course there was kind of a range of how mature they were. So when we applied compost that were a bit less mature, they definitely tied up a lot of nitrogen right away because just they're more, the carbon is more reactive. Daniel's lab right now is doing some more work on looking at specifically crop residues. But my impression is that the general rule of look at the percent nitrogen, look at the C to N ratio, and it will give you a pretty good idea, will still hold.
Danielle, any comment?
Add anything to that, Danielle?
Yes, so we did an incubation. We were wondering, does it make a difference if you leave the crop residues on the soil surface and let it dry, or if you incorporate it when it's moist? And we did it with broccoli residue and tomato residue. And what we found is that if you dry it first or incorporate it immediately, has an effect on nitrogen turnover during the first week or two. And after that, we didn't see any difference anymore. And we also looked at different soil moisture contents, and in the long term, the moisture content of the soil had a much bigger effect than the initial residue, the moisture in the residue at the beginning. So it's a relatively short-term effect.
Okay, thank you. Right, here another question. Why do we rarely see the percent organic matter in the soil increase even after years of adding compost and organic fertilizers? Is that for Danielle or Margaret, or any answer?
One thing I can say, if you have a soil organic matter content of, let's say, 2%, and you measure the total nitrogen in the soil organic matter in the top foot, it can be up to 2 tons of nitrogen in that soil organic matter with 2% soil organic matter. So you have a very big pool of nitrogen and carbon in the soil already. So that means you have to add over a long time to really see a difference, an increase in soil organic matter content, just because it's so big.
Yeah, right. Margaret?
Yeah, I mean, I think to build off of what Daniel said, you know, we can also think about our soil organic matter pool is quite active for many years after adding material. And so there's a lot of turnover happening. So all this crop residue and compost is being utilized by the microbes and then utilized by our plants. So, and it seems like once it, you know, after several years really shifts into more stable forms of organic matter, that's where maybe those increases can start to be measurable. But that's after several years, and also after the crops have taken what they are going to use, and et cetera, the sort of active cycle that's happening.
Okay. Our next question to Richard, I guess, on the subject of applying manure compost to almond orchard who are on no-till systems. Based on Richard's work on the Central Coast with nitrogen availability from surface versus incorporated, how much more nitrogen is burning off? I know the Central Valley has higher temperatures swings too. Richard? Is Richard still there? Maybe not. All right. Someone else, like Patricia. Do you have any comment?
I couldn't give numbers.
Okay.
In general, like the things that will make a difference are if it's being irrigated into the soil, you'll lose less just because as it's or some labile carbon compounds can actually be washed into the soil, and then once it's reacting with the soil, there's a lot less potential to volatilize. Another thing that makes a big difference is the soil pH. So the more alkaline the soil is, the more likely that you are to lose it. But I couldn't give exact numbers off the top of my head.
Yeah, this is no-till systems works, and then there's a lot of work on, you know, like Midwest or so forth. But in California, yeah, I think we need to do more this kind of work. There's some literatures how that's no-till system affect nitrogen mineralization. So if you're interested in, I can send some references. So email me. Okay, another question here. Compost application is always limited by practical delivery to the fields beds. Are there any new compost technologies to make application more practical, whether this be machinery for application or type of compost? Margaret.
That's an excellent question, and I don't have a good answer for you. You know, maybe the timing of application could be a way to think about easing any of the logistics, but that's, you know, neither one of your ideas. Yeah, it's a hard one. I don't have a good answer for you. Maybe
Anybody?
maybe you.
Right, anybody, any comments? Okay. All right. Well, then move on to the next one. Yeah, we still have some time. A couple more questions. Is there any benefit by adding amino acid products or acidifying soil?
For the, or Margaret, do you want to comment on that, or should I?
Go ahead, Patricia.
Okay, well, for amino acid products, those will definitely be very, very quickly available. Amino acids are broken down extremely quickly in the soil, and under some conditions, plants can even take them up without breakdown. So as a nitrogen source, it will be very, very rapid. And it won't leave much carbon behind. That's of course always the trade-off between building soil organic matter and providing nitrogen. So very, very rapidly available fertilizer. For acidifying the soil, that's a super, super interesting question. A lot of it depends on your initial soil pH and on the crop. But a lot of micronutrients especially are more available at slightly more acid pH, and some crops like tomatoes just prefer a slightly acid pH. I guess part of it depends on what you mean by benefit, but there's most biological processes tend to occur best at a neutral pH. So acidifying too much, probably not a good idea, but then every crop has its preference as well.
Right.
So not sure if that answers your question about the acidification.
Great, thank you, Patricia. All right, any other questions? We still have like time for a couple more questions, I guess. And then Rachel's gonna show some CCA credit things. So any more questions? All right. Then, well, again, thank you very much for the all speakers. Excellent talks. And well, this concludes the second session of the workshop on nitrogen management in organic systems. And we're gonna have another one, last one, next week, same time at 9 a.m. and in which myself and Richard Smith gonna talk about like kind of wrap up session, put everything together, the things we learned last week and today to actual cropping system, such as strawberries, organic strawberries, and organic vegetables. And at the end, I'm going to talk about a little bit of, you know, kind of frontiers of nitrogen mineralization or availability studies. I'm going to introduce some advanced studies on nitrogen management related topics. Where is science are right now, that kind of topics. So with that, thank you very much for attending. And if you have any questions, feel free to reach us. And I think this project presentations are recorded. So it's going to be available at the Monterey County UC Cooperative Extension website in the future. And then here's the Rachel showing the some CCA credit QA code and so forth. So please use this. Rachel, any announcement?
You've covered everything. Thank you. And hopefully we will see you all next Tuesday, same time, same place, same Zoom link.
Thank you very much, everybody.